Hello students, welcome to Jiu class. So today we'll be going to chapter 7, which is biodiversity. So chapter 7 has a lot of mini chapters. So this is the first chapter. We will deal about all of the kingdoms of life. So there are 5 kingdoms of living beings. Until 18th century, people only know about 2 things. One is animals, one is plants. So either plant or an animal. Plants, what they do is that they form photosynthesis. Animals, they find their own food. But however, there are animals that behave like plants and plants that behave like animals. And they are different organisms which are structurally different, but they look similar. For example, cyanobacteria and blue-green algae. Euglena is this organism which are both animalistic and also able to do photosynthesis. So are they animal or are they plant? Okay, so there's a new way of classification proposed by R.H. Whitaker in 1959. He classifies organisms based on 2 criteria. One is level of organization and the other is method of nutrition. Okay, so according to his classification, if organisms can be either prokaryotic or eukaryotic. Eukaryotic can be either simple cells or multicellular. Okay, and for multicellular, you have autotroph and heterotroph. So here is the 5 kingdom system of classification. So you have Monera, Protista, the plants, animals, and fungi. Updated, we also have archaea, not just bacteria. So, bacteria versus archaea. Prokaryotes do not have any nucleus. But bacteria and archaea, both of these don't have nucleus, but they are a little bit different. For example, DNA, RNA, they are different. Genes are much more similar to eukaryotes. Cell wall, they lack of bacterial glycan. And habitat, archaea are only found in extreme conditions. So first of all, before going to bacteria, we learn about viruses. Viruses, they're not exactly classified as living because they do not show any characteristics of a living organism. Yeah, they do not eat, they do not breathe, they do not even have organelles, they do not even have any metabolism, they cannot multiply by themselves, so they are not actually part of the living kingdom. For viruses, inside of the host, they're called virus. Outside here, they're called viron. Smaller than bacteria, 100 to 300 nanometer. So a typical virus will have 2 parts, genetic material and protein capsule. So this virus, they are not alive because outside of the cell, they do not do anything, but inside of the cell, they're able to multiply. Classification is based on the type of nucleic acid that they have and the shape of protein capsule. Protein codes will have different type of shapes, may have another layer, the lipid envelope surrounding the capsid. For example, poliovirus, they're round. Adenovirus, they look like an icosahedron. Same with herpesvirus, but herpesvirus actually has a lipid envelope. Bacteriophage looks like a spider. Mumps virus, they are round and large. Vaccinia, they are squarish. So, bacteriophage, these are viruses that consume bacteria. They have to infect bacteria. They are obligate cellular parasites. So, how do they infect? They get into the cell and they hijack the host cell to make more parts of itself. So, their parts consist of head or capsid, which contain nucleic acid. So, this head will be covered and protecting the nucleic acid. Tail, these are the hollow tubes in which nucleic acid pass through during infection, and they are surrounded by sheath that can contract during infection. So, for binding to the bacterial cell, the base plate and tail fiber are involved. So, the bacteriophage can infect hosts in 2 ways. One, they insert their DNA into the host DNA, or they can destroy the bacteria in the process. So, there are 2 cycles, lytic and lysogenic. So, here is how the virus would attach. So, it lands, builds, contract their tail and penetrate before injecting their DNA. So for attachment, virus land on cell membrane by either mechanical force or receptor. If there is no attachment, there is no infection. And then entry. What will be entering will be the phage DNA, and when this happens, host DNA will be degraded. Okay. And then after the cell has been hijacked, it will be directed to perform biosynthesis, which is the creation of viral genomes and proteins of the virus by the bacteria itself, by transcription and translation. The assembly, this is when all of the parts that are created, they will combine together to make the phage. And then release. All of the viruses will be released from the body of the cell due to the enzyme that is called lysis, okay, and entry of fluid. The whole cell bursts, releasing 200 to 200 phages, and when they are free, they are able to infect more and more bacteria to continue the cycle. So here is the whole lytic cycle. It ends in the whole cell exploding and releasing the completed viruses. For the lysogenic cycle, initial stages are similar to lytic cycle. Instead of viral genetic material taking over, the viral genetic material actually inserts itself into the DNA of the bacteria and stays there. So whenever the cell divides, the bacterial DNA will also divide. So it can continue indefinitely until it triggers the lytic cycle. But usually the host cells are unaffected. So here is cycle. The cell will prophase, phase DNA secularizes, and then it can go to lytic cycle or lysogenic cycle. So when this prophage or the phage DNA is actually inside of the DNA of the host cell, they will divide whenever the host cell divides. So comparison, lytic cycle will cause destruction of host DNA, production of new phages, lysis of host cell causes release of progeny phages, and genome integrates to bacterial chromosome as prophage, which is replicated and passed on to daughter cells. And this can be induced to leave this cycle to go to the lytic cycle instead. Importance of viruses are actually quite important because in biotechnology we use virus to manipulate artificially genetic material from one organism to another and to transfer them. Gene therapy are used to fix somebody's faulty genes using viruses. So you get the correct gene injected into a sick person so that they can repair their genetics. Okay? Or it can be used to inject suicide genes into cancer cells so that the cancer cell can be self-destruct. Alright, now we move to kingdom Monera or bacteria. So characteristics of kingdom Monera, all of them do not have any nucleus or membrane organelles. So prokaryotes are much smaller than the smallest eukaryotes. All of them are unicellular, and now there are two kingdoms, archaebacteria and eubacteria. Archaebacteria, archae means ancient. Evolution considers them as the first organism, and they can live where no others can. So this means that they live in areas which are extremely hot or cold or salty. Okay, they live in the area which are dangerous to any other organisms. So the first side is thermoacidophiles. So they live hot and acidic environments. Many of them are chemosynthetic, meaning that they can use the chemicals to create organic material so that they can consume them. So 40 years ago, this molten door to hell called Darvaza crater opened in Turkmenistan, and until now it is still burning. So a Canadian explorer, George Cornish, he became the first person to descend into this fiery pit to find life. And what he found was there's a lot of bacteria living in the bottom. They are very comfortable in living in the high temperatures. And these bacteria are not found in the soil surrounding the crater. So this actually gives hope that there are life in the solar system or beyond the solar system, because beyond the solar system, the conditions are extreme, too cold, too hot, no air at all. But still, the Davasa crater bacteria shows that they can live in those type of environments. So archaebacteria number two, extreme halophiles. These are the salt lovers. They like to live in the area which have a lot of salt that can actually kill other organisms due to the hyperosmolarity. Okay, they'll be hypertonic. They will suck the moisture out of them. But these halophiles can live there. Okay, they use salt to generate ATP. So now for eubacteria, it's classified into two ways, shape and stain. There are three different shapes and more, and two different stains. Three shapes. The first shape is coccus. Okay, they are rounded shape, either dark coccus. Two The Staphylococcus are clusters, Astrococcus are the strings. Rod shape, we have the bacillus. So you have Diplobacillus, which is 2, Streptobacillus, which are strings. Spirillum, they look like spirals. So for stain, there are two reactions to stains. You have the gram stain. So it's either gram positive or gram negative. So gram negative will be pink color, but gram positive will be purple color. So for gram positive, they are purple in color. Okay, their simple cell wall and antibiotics are effective. But gram negative, they are pink because they do not grab the gram stain. They absorb the secondary pink stain instead, and the complex cell wall makes them very resistant against antibiotic. So according to Alexander Fleming in 1928, okay, he is the first person who found something called antibiotic. So antibiotics were found by accident because he, who is a Scottish bacteriologist, he was growing Staphylococcus bacteria in a dish, and somehow it got contaminated by the penicillium mold. And mold inhibited the bacteria. So if you can see in this picture, in the middle there is a large patch of mold, and around the mold there is a clear area with no bacteria or no growth at all. So meaning that those are area which are clean, and something in it actually has killed the bacteria. So it is found out that the penicillium mold created a toxin that inhibits formation of bacterial cell wall. Okay, you can see this picture. On top of that, you can see the penicillium mold, and around it there's an empty space. And after the empty space, you can see colonies of bacteria. Okay, okay, you can see that the penicillium mold. If you zoom in, they are just like just fungus. Okay, oxygen requirement for microbial growth. Some of them are obligates, mean that they need to have oxygen so that they can live. Okay, facultative anaerobes, they are able to use oxygen when available, but they do not require it. Okay, like Escherichia coli. So the third oxygen requirement for microbial growth will be obligate anaerobes. So obligate anaerobes mean that they have to live in areas with no oxygen at all. For example, Coccidium tetani. So in photosynthesis, okay, photosynthesis is not just done by plants but also bacteria, especially cyanobacteria. So they are able to create CO2. They are able to create O2 by photosynthesis, and the availability of O2 would give the advance of aerobic organisms. Bacteria in nature, you have the rhizobacteria. These grow in roots of soybean, alfalfa. Then they convert atmospheric N2 or nitrogen gas into usable form for plants. So decomposers, these will break down matter into similar substances, and a lot of nutrients are released into the soil. In the belly of the termite, you have the zoa Mastagopera. Okay, they are symbiotic with the termites. Right, so that's the end of the bacteria and Monera chapter. Thank you.